Okay, I'm going to do a video lecture on the cytoskeleton, so I'm going to do briefly, I'm not going to do all of them, but I'm going to kind of compare and contrast or go over the intermediate filaments, the microtubules, and the actin filaments, talk about how they make the filament, and then some of their properties and what regulates their dynamics. And so hopefully it'll be a brief review, try to keep it brief, of the different types of cytoskeletal filaments and how they operate. All right, so remember, the three types of cytoskeletal filaments are the actin filaments. They're also called the microfilaments because they're roughly 7 nanometers in diameter. The microtubules are the largest filament. They're an actual tubule. They're about 25 nanometers in diameter. And the intermediate filaments are called intermediate because they're in between the two sizes. They're roughly 10 nanometers in diameter. And this is a fluorescent image of each type of filament. And just remember that these exist simultaneously in the cell. All three filament systems are inside the cell, all doing different things at the same time. So we'll start with intermediate filaments. And one thing to keep in mind is their most important function is to provide what's called tensile strength. So this is the ability for cells to withstand pulling and pushing and gives them some kind of interior type of structure and rigidity. So if you didn't have intermediate filament within the cells, remember if you take the cells here and you stretch them apart from each other, ultimately what will happen is the cells will separate from each other and they'll actually tear apart. But with intermediate filaments, they help link cells to cells and bridge cells over and over again. And by linking them to each other, and by the filaments being linked to each other between cells, is when you pull on the cells, they resist that pulling. And so remember down here, what you see is when you look at a sheet of cells connected to each other, you actually see the intermediate filaments bridging the two cell junctions. And they do so through a structure called the desmosome. And the desmosome is a specialized kind of like weld-like structure that connects the intermediate filaments of one cell to the intermediate filaments of an adjacent cell here. And therefore, when you pull on these two cells apart from each other, they resist pulling and they won't be pulled apart. So that's their main function. So there are 4 types of intermediate filament proteins. There are 4 different classes in cells. And all I want you to know is where you'll find these different types of classes. And remember, they all work the same way and they all function the same, they all have the same function. So inside the nucleus of cells, we talked about nuclear laminins. And these are in all animal cells, and these coat the inside of the nuclear envelope and give rigidity to the nucleus. Within the cytoplasm of cells, you have neurofilaments, which are in nerve cells. You have vimentin, which is the name of the intermediate filament protein that's found in connective tissue, muscle cells, and glial cells. And glial cells are kind of support cells of the nervous tissue. And then you have keratins, which are found in the epithelium. Okay, so one thing to keep in mind, these are 4 different intermediate filament proteins. They're made from 4 different genes, but they all make intermediate filaments. So they all function the same way and form the same type of filament structure, even though they're different proteins. And so in all cells, you'll have the nuclear laminins, and then in nerve cells, you'll have the neurofilaments, and then say in epithelial, you'll have keratins. So in reality, in all cells, they'll at least have 2 types of intermediate filaments in them because they all have nuclear laminins to support the nucleus. So here's how intermediate filaments form, and we'll focus on the very top first. And so intermediate filaments make ultimately a very strong rope-like structure, and they do so. Here's the first monomer of the protein. Okay, so this is, say, vimentin. This is one polypeptide. Here's your N-terminus, and here's your C-terminus over here. And this is a monomer. And so you have these globular ends that look like little balls. And then in the middle is an alpha helix. Okay, that strings down there. And so 2 alpha helices, or 2 different intermediate filament proteins, will come together and form a coiled coil dimer. And these dimers form in a parallel fashion so that both the N-termini are facing one end. Okay, and both the C-termini are facing the other end. So this is the dimer form. Those dimers will then undergo a formation of a tetramer. So 2 of these dimers, so let's say times 2, 2 dimers will come together and they'll do so in what's called a staggered tetramer. and they'll do it in an anti-parallel fashion. Sorry for my handwriting. Anti-parallel. This is really important because what happens is now that it's anti-parallel at the tetramer stage, one end of the filament, or part of the filament, has only an N-terminus, and the other end has an N-terminus. Therefore, we've lost polarity, so no polarity. All right, and this isn't not necessarily important in intermediate filaments, but it's important in all the filament structures because if you remember, for microtubules and actin, polarity is really important for how they polymerize. So anyway, you have your staggered tetramer, and now these tetramers will pack together end to end and side by side, and they'll essentially form a twisting rope-like filament that you'll see here that's roughly 10 nanometers in diameter, and this is the intermediate filament. Okay, so it starts up here with the monomer forming the dimer. Two dimers form a staggered tetramer that's anti-parallel. The tetramers will pack together end to end and side by side, and they'll form the twisted rope-like intermediate filament. So because there's no polarity, intermediate filaments don't really have any dynamic polymerization like the microtubules and actin filaments do. Once they're made, they're very stable because they're meant to provide rigidity and structure for the cell. So those are intermediate filaments. Remember, their role is mechanical strength, and just know how they come together to form the filament stage. Next, we're going to move on to microtubules, and these are the largest form of the cytoskeletal filaments. And you'll find microtubules in all animal cells, and they're important for a variety of reasons, and they form a variety of different structures. So in an interphase cell, so what this means is just resting, or a cell that isn't undergoing cell division or isn't specialized or anything, the microtubules form a network of tubes, of little tracts that are used for organelle and vesicle movement. And we're not going to do it here, but in the lecture and class we went over the different type of microtubule motor proteins, dynein and kinesin, and those are important for moving the vesicles and organelles back and forth throughout the cell. So in a resting interphase cell, you'll look and you'll see a lot of organelle movement back and forth throughout the microtubules. In a dividing cell, microtubules are really important because they make up the entire mitotic spindle. So they're responsible for separation of the chromosomes and cell division. So if you inhibit microtubules during cell division, you'll inhibit cell division. And then also in specialized cells, such as your epithelial cells, you have cilia, or ciliated cells, all right, that are necessary for movement of particles. In things like sperm, you have flagella that are necessary for movement, and those are all made up of a special structure that are made up of microtubules called the flagellum 9+2 microtubule array. So we're not going to again go into those structures, but again, those are formed by microtubules. So microtubules do all three of these things, depending on what the cell is doing or what type of cell it is. So here's microtubules and how they form. So let's start with the monomer subunit. And the monomer subunit of the microtubule is called a tubulin heterodimer, and it's shown here. And it's a heterodimer, which means it's two different polypeptides. One is called alpha tubulin, right there, and one is called beta tubulin. So here's the alpha tubulin, here's the beta tubulin. And typically in the cell, as a monomer, they exist as a dimer. All right? So most of the time when you see unpolymerized microtubule or polypeptides, you'll see the tubulin heterodimer. So this is what makes up the overall microtubule. Two things to keep in mind with this heterodimer is they both can bind GTP. Okay, so you'll have GTP bound to them, but only the beta subunit hydrolyzes GTP. Okay, so remember hydrolysis of GTP is GTP goes to GDP and PI. All right, so only the beta subunit does this. The reason the alpha is always stuck in the GTP bound form is just because of the nature of the heterodimer. It prevents the alpha from hydrolyzing it. So in reality, they both can hydrolyze GTP, but inside the cell, only the beta unit does. And that's important when we get into how they polymerize. So here's how the microtubules form. Microtubules have polarity. The reason being is they form end-to-end with the heterodimer. So you have one heterodimer here, okay, You have another heterodimer that stacks on top of it, and the beta subunit will add to the alpha subunit, and over again. So no matter how long your filament is, on one end you're going to have the exposed beta subunit, and that's what we call the plus end. And on another end, you're going to have the exposed alpha subunit, and that's what we call the minus end. Okay, so because of the way that the heterodimer polymerizes, you have polarity. And this is important for how it regulates itself. So when the microtubule grows, it'll grow by addition at the plus end. So more tubulin subunits will add at this end, okay, and you don't get addition to this end. All right, so the plus end means that that's the side of the filament that's going to grow. So these will continue to polymerize, and they'll make a chain of tubulin subunits. Okay, and this chain right here is called a protofilament. Protofilament. Okay, and 13 protofilaments will join side by side and wrap around each other to form a microtubule. So you see here, if you count these dots up, there's 13 of them. And they wrap around each other, and so inside of here is the lumen of the microtubule. It's hollow. There's fluid inside of there, and so it forms a tubule. Okay, and so from this end of the microtubules, you can now get increased addition of the heterodimers on each protofilament, and you can grow the microtubule. So that's how they polymerize, and they start with the heterodimer, form the protofilament. The protofilaments wrap around each other to form a tubule, and this tubule can then polymerize off the plus end. In an interphase cell, or in all cells, microtubules don't just grow in the cytoplasm. They're typically started by some kind of nucleating factor. Okay, so inside a cell, you have a very, not large, but a very rich in protein type of organelle called the centrosome. It's not really an organelle because it's not membrane bound, but it's a complex of proteins that are important for polymerizing microtubules. And so the centrosome, this green sphere you see here, is just proteins. It's not a membrane or anything like that. But inside of all these proteins, you have what are called centrioles, and you have a pair of them. So you have a pair of centrioles, and these centrioles are actually short little microtubule pieces. And so they help stabilize the centrosome structure. Okay? So they're small arrays of microtubules. And like I said, they're important for stabilizing the entire centrosome organization and structure. So what does the centrosome do? The centrosome is important for stimulating the polymerization of microtubules from it. And so the centrosome is kind of the starting point for microtubules, and all microtubules will radiate from the centrosome, as you can see here. And remember, they only polymerize off the plus end. So therefore, when you look at an array of microtubules coming off of a centrosome up here, okay, you see that all of them have their plus ends exposed. Okay, so they're all going to be polymerizing from the plus end off of the centrosome. And the reason for that is part of the centrosome is a structure called the gamma tubulin ring complex. And this is called a nucleating site. A nucleating site just means a site that starts polymerization of the microtubule. And the gamma tubulin ring complex is a series of proteins that form a ring. And this ring looks like the plus end of a microtubule over here. Okay, so it's not necessarily, it is tubulin. Okay, it's a different type of tubulin. It's called gamma tubulin. And it looks like the plus end. And so when you have free tubulin heterodimers around, all right, these are all going to add to this, what they think is the plus end, and the microtubule is going to grow. Okay, so you get your plus end there. So that's called the gamma tubulin ring complex. It's the nucleation site for microtubules, and they're all part of the centrosome, which is stabilized by the centrioles. Okay, these centriole pairs inside of there. So if you didn't have the centrosome in a cell, you couldn't nucleate microtubule polymerization, and you wouldn't be able to get any microtubule formation. We went over the fact that the microtubules can hydrolyze GTP, and so what happens is the beta tubulin subunit with bound GTP wants to add to the plus end. Okay, so we'll draw briefly over here. If you have alpha beta, and this has GDP on it, and you have alpha, beta, and this has GTP, okay? The incoming subunit, the alpha, is more attracted to this beta GTP, okay, than it is to the beta GDP, okay? And we're not going to get into why, but it's a structural thing. So the alpha is going to add to the beta GTP, but it won't add to a beta subunit that has GDP. All right, so what this means is that if you polymerize the microtubule, right, and you start polymerizing it, once these heterodimers add to the microtubule, it stimulates their hydrolysis of GTP. Now the hydrolysis of GTP takes a few seconds. It's not immediate. It's pretty quick, but it's not immediate. And so what happens is if you polymerize really fast and you add a lot of subunits on before you lose the GTP, you have a region of the microtubule called the GTP cap. Okay? And this GTP cap is going to recruit more heterodimers to add to it, and you'll get growth of the microtubule. So essentially, if the rate of polymerization is greater than the rate of GTP hydrolysis, the microtubule will grow because, and this is important, because it contains a GTP cap. All right? The rate's faster than hydrolysis, so that the end of the microtubule always has GTP on it, this GTP cap. However, if the rate of hydrolysis is faster than the rate of polymerization, so if for some reason, let's say you run out of heterodimers at the end and all of a sudden GTP hydrolysis, right, catches up to the end of the microtubule, you will get depolymerization. This thing becomes unstable and it completely falls apart, and the microtubule will shrink in a process that is called dynamic instability. Okay? So again, if you maintain the GTP cap, and the way to maintain the GTP cap is by polymerizing faster than you're hydrolyzing, then you'll grow the microtubule. If you lose the GTP cap because the hydrolysis rate increased faster than the polymerization rate, then the microtubule will shrink. So if you memorize these two statements, you'll be good. So that's the whole process of dynamic instability. So these microtubules are constantly growing and shrinking within the cell. Now, it doesn't always have to happen that way. There are other proteins in the cell that can control microtubule dynamic instability, and they can stabilize a microtubule so that it's not constantly growing and shrinking. So those are microtubules. And I didn't do everything in that lecture, so please go back to that lecture and review what we went over with the rest of the microtubule structures. But that's essentially how they form and what regulates their polymerization. And lastly, we're going to finish with actin filaments, which are the microfilaments. And these are very important too. One of their main reasons they're important is they're really universal to the cell, just kind of like microtubules are. They do a lot of things. So for example, they form the microvilli, and these were important in the intestinal epithelial cells that we talked about during protein transport. They form contractile bundles, so not only are these important for cell migration, but they're really important for muscle contraction, obviously. They form dynamic protrusions down here in C, which is important or really crucial for cell migration. And then they form ultimately the contractile ring, which is a small ring-like structure. It's almost like a mini muscle that contracts and actually pinches the two daughter cells from each other during cell division, and is the final step in cell division of cytokinesis. Okay, so they form these four major structures, and they do a lot of other things as well, so they're also very important. All right, so remember, actin filaments are also called microfilaments because they're the smallest filament in size. They're roughly 7 nanometers. And this filament is actually 2 filaments in one. They're 2 filaments of actin monomers that are twisted around each other. So they form kind of like a helical pattern. So if you took 2 beads on a string and you kind of wrap them around each other, that's what an actin filament would look like. So again, they're made up of monomers, just like the microtubules are, and just like the intermediate filament proteins are. And the monomer is called globular actin, or G-actin. And when put into filament form, we call it F-actin for filamentous actin. Okay? So we're not going to go over the crystal structure of actin, but it does look different on one side than the other, so therefore actin has polarity. So actin has polarity where one end of the molecule looks different than the other end. So let's go over here to the structure. You can see up here what's called the minus end. Kind of has these little protrusions out here and this little pocket. That's much different than the plus end that has kind of a flat structure here. And so because of the way the actin filament looks, it has polarity. And so we call the end that adds new monomers, that's called the plus end, just like microtubules. They polymerize at the plus end, and they lose actin monomers at the minus end. Okay, so that's the polarity. So both actin and microtubules have polarity. Intermediate filaments do not have polarity, and the reason why intermediate filaments don't is because of the way they make a filament. So go back and review that if you're still confused on why they lack polarity. So here's how actin filaments can polymerize, and it's very similar to microtubules, just with a small couple caveats. First, actin binds and hydrolyzes ATP. Remember, microtubules, so right, MTS, microtubules bind and hydrolyze, GTP. Okay, but the same is both true. They bind to ATP, and then when they move on to the filament, when the G-actin binds to the filament here, it stimulates their hydrolysis, which takes a few seconds, and they hydrolyze ATP, release a phosphate, and now you have ADP containing actin. Okay, so very similar to microtubules. Like tubulin, okay, ATP-bound actin has a higher affinity to form a filament. So what that means is that if you have ATP actin here, it's going to want to form the filament more than ADP wants to. Okay, so this one doesn't want to form a filament, it doesn't like it. The ATP-bound actin does. So what's going to happen in order for this monomer to get back onto the filament again, it's going to have to somehow exchange ADP for a new ATP so that it can add to the filament. All right. ADP actin, however, because it doesn't like the filament, is more likely to disassemble. So what this means is that you're going to have addition at the plus end, so remember this is the plus end of the filament, of ATP actin, and you're going to have subtraction at the minus end of ADP actin. And what will happen is if you watch this filament over time, as these add, these will subtract, and this filament is actually going to move in this direction. Okay, and this is what we call treadmilling. Right, so the property, sorry for the handwriting, treadmilling, the property of the actin filaments, the way they polymerize. And if you watch this over time, it actually moves forward. So there's some animations on YouTube you can probably find of this happening. But just remember, the ATP actin binds to the plus end. When it binds to the filament, hydrolyzes ATP, becomes ADP-bound actin, and at the minus end has less affinity for the filament and therefore will disassemble at that minus end. All right, so we talked about 3 different types of structures that the cells make in order to crawl. We went over the process of cell migration. I'm not going to do it here. But 3 different structures that are all dependent upon actin: the filopodium, the lamellipodium, and the contractile bundles. And I just briefly talked about one actin accessory protein that goes with each one of these. So we'll start with the filopodium. In the filopodium, let me go back, I'm sorry. The filopodium are these small finger-like projections right here of the cell. And they're kind of meant for sensing the environment and sensing the rigidity of the substrate they're crawling on. They have a lot of other functions as well. And they're very straight bundles of actin, as you can see here. Okay, they're very straight and rigid. Then you have the lamellipodium, which is a very broad sheet-like structure behind the front edge of the cell. Okay, this is kind of a broad sheet-like structure, all made of actin. And it's very branched actin. So it's a lot of actin crisscrossing back and forth with each other. And it makes a very high amount of actin inside that pushes the plasma membrane forward. That's called the lamellipodium. And then in the rear of the cell, you have contractile bundles. And these are a series of actin filaments that are anti-parallel to each other. And we'll talk about it, but there's myosin in here, all right, that's putting force on these actin filaments and pulling these actin filaments to each other and thereby contracting the cell forward to pull the rest of the cell forward as it's moving. So formins are one of the actin accessory proteins that are important in forming filopodia. So formins, you can just think formins, filopodia. All right, and what formins do is they bind to the plus end of the actin filament, and they basically help or promote the addition of G-actin, right, to the filament. And they do so to prevent actin from becoming branched. Remember, filopodia, you want to form very long finger-like projections. You want very long, unbranched actin filaments to push those fingers out. And so formins help accomplish that. And so they bind to the front of the actin filament, G-actin comes in and binds, the foramen changes shape, allows another G-actin subunit to come bind here, and then it changes shape again, allowing for another actin subunit to come here. And slowly will allow the actin filament to grow. It's not really slow, actually, it's very fast. In the lamellipodia, remember, it's not like foramens. You need a very broad, so if we draw a cell here, you want a very broad sheet-like structure of the front of the cell that's all pushing out forward for the cell to move in one direction. And so in order to accomplish that, the cell makes a very highly branched network of actin filaments that you can see here. It's very hard to see, but each one of these is an actin filament, very complex and branched. That branching is accomplished by a complex of proteins called the ARP2/3 complex. And so what happens is the ARP2/3 complex binds to the side of an existing actin filament, and the ARP2/3 complex looks like the plus end of an actin filament. And the reason being is ARP stands for actin-related protein 2 and 3, which means that these proteins actually are like actin themselves. But they look like the plus end of a filament, and therefore they nucleate polymerization off the side of an existing actin filament. So as a filament is growing, ARP2/3 binds to the side, and you get branching. And then as it keeps on going, ARP2/3 binds to another side, and you get branching. And as this filament grows, you get ARP2/3 binding, and you get more branching. And you continue this over and over again, and you make very large branch-like structures. So as these new filaments are growing, ARP2/3 is going to bind here at the very front, create a new branch that's going to grow, and ARP2/3 will bind, create a new branch over and over again. Okay? And one thing that's interesting about ARP2/3 is the way that the structure related that as it binds to the side of a filament and forms a new filament, it's always going to do so at a 70-degree angle. 70-degree angle. Okay, so it's making these 70-degree angles over and over again, and over time, this produces a very rich, complex network of actin filaments that forms this lamellipodia. And then lastly, we talked about the contractile bundles, and we really went into the muscle and how the muscle works. But the contractile bundles are actin filaments that are anti-parallel to each other. So you can see here is one actin filament, and here's the plus end of one, and here's an actin filament and the plus end of another. And in between the two actin filaments is a myosin II bipolar filament. And remember, myosin II bipolar filament is a bunch of myosin II motor proteins that are all facing each other. Okay, so these are kind of anti-parallel, not anti-parallel, they're end-to-end facing each other. So what that means is, remember, myosin II is a actin motor protein that migrates towards the plus end of actin. So this head, okay, this myosin II head, is going to want to go in this direction. And this myosin II head is going to want to go in this direction. Well, let's say that this filament here is linked somehow to the cell. So it's stable and it's rigid and it can't move. When these heads act on these filaments, they're going to pull these filaments in this direction, and they're going to pull the filament in this direction. And what's going to happen is, overall, these two filaments will slide closer to each other and form a contractile structure. Okay? And that's the basis of contraction. So in order for contraction to happen, first you need a bipolar filament of myosin, and then second, you need anti-parallel actin filaments. If these filaments were facing the same direction, you wouldn't get this result. And you can practice drawing it and see for yourself. But ultimately, these are this setup here allows for contraction. And so this basis that happens inside the cell during cell migration is the exact same concept for how muscles contract and the organization of skeletal muscle. And so here we went over briefly the organization of skeletal muscle. And what I want you to understand is that there's a myosin bipolar filament. In muscle, it's a lot bigger, and there's a lot more myosin molecules, but it's the same concept. And this myosin bipolar filament, it's not shown here, but it's anchored at a structure of the sarcomere called a Z-disc. Remember, the sarcomere is the basic contractile unit of muscle. So these are anchored in place. The myosin bipolar filament doesn't move. And what you have at the Z-disc are the plus end of the actin filaments here. So plus end, plus end, plus end. This is the minus end of this actin filament. This actin filament, this is the minus end, and this is the plus end in the Z-disc, plus end here, plus end here. So these myosin molecules migrate towards the plus end, so they're going to want to go this way, and these are going to want to go this way, because they're anchored in place, the force they produce is actually going to push the filaments in this direction, pull, and it's going to pull the filaments in this direction. And by doing this over and over again in this kind of arrangement, you get a sarcomere that contracts when the myosin molecules move along the actin filaments. Okay? And so relaxation involves contraction of an opposing muscle. You also need relaxation of the myosin heads. They need to unbind the actin so it can go back to normal. So remember, this is called the sarcomere, the basic contractile unit of the muscle. You have the Z-disc where the plus ends of actin are anchored, the myosin bipolar filament that's stabilized within the sarcomere and moves towards the plus end of the actin filaments and therefore pulls the two Z-discs closer to each other. So if you look here at relaxation, the Z-discs are this far apart, and then at contraction, the Z-discs now are this far apart. So when you contract the sarcomere, the Z-discs become closer together. The actin filaments become closer together as well. Okay? So that's kind of the basis of muscle contraction. Again, it's dependent upon actin and myosin working together, and that's the fundamental concept of how all kind of contraction works in the cell. All right, so I hope this helped you out. These were the brief different cytoskeletal filaments. You should go over what they do, how they form, what's their monomer, how are they regulated from polymerization, things like that. Understand how they become a filament, especially in microtubules and actin. Understand the concept of hydrolysis of GTP and ATP and how that regulates its formation.